Hi everyone. So the title of my proposal is basically investigating the structural basis of strain-specific inhibition of RIG-I by non-structural protein 1 of influenza virus. So um, influenza is a disease, is a major burden on human health as well as economics, and it causes seasonal epidemics and occasional pandemics. For example, the last pandemic, 1918 influenza pandemic, resulted in the death of 50 million people worldwide, and this chart just shows that each year, on an average, there are 30,000 deaths in the United States alone. So, understanding how the influenza virus works and inhibits the host defense system is really critical. So, now coming to the influenza virus, it is a negative sense single-stranded RNA virus and has a host of different, range of different hosts. For example, humans, birds, pigs, and whatnot. Its genome is segmented RNA virus, segmented RNA, which codes many different proteins, structural proteins. On the surface, you will know that there are hemagglutinin proteins, neuraminidase proteins, which help the virus gain entry into the cells as well as the exit from the cell. But one of the key proteins is the non-structural protein 1, which is the main virulence factor of the virus. And the NS1 protein inhibits host innate immune response using variety of strategies. For example, it interacts one of the key viral sensor in the cytoplasm known as RIG-I to inhibit the signaling pathway. It also interacts a host protein CPSF30, which is a polyadenylation factor, to inhibit the host gene expression. It blocks activation of interferon stimulatory genes. Also regulates apoptosis. So it's a multitasking viral antagonist, which just a single protein, it antagonizes the host immune response. So, but in this proposal, we'll focus on one of the pathways, the RIG-I antiviral signaling pathway. So, as I mentioned, RIG-I is a cytoplasmic viral sensor. In the uninfected cell, it is in the inactive state. After it detects viral RNA, it becomes active, and after activation, it interacts with a downstream protein known as mitochondrial antiviral signaling protein on the mitochondrial membrane to form a complex, and this formation of this complex actually induces the interferons, which are basically small molecules which alert the neighboring cells as well as the same cell itself for mounting defense system. So, as you would see, like NS1 can inhibit this pathway in multiple levels. For example, it can inhibit the detection of viral RNA by the RIG-I itself. It can also inhibit by interaction with RIG-I to inhibit the downstream signaling pathway. Okay, so these two proteins of our interest, one is the viral protein NS1, has a RNA binding domain, internal RNA binding domain, also has a C-terminal effector domain, and both of these domains interact with diverse host proteins. And the host protein, which is RIG-I, has two internal card domain, card 1 and card 2, and a helicase domain and a C-terminal domain. So now let's see schematic of how NS1 inhibits the RIG-I pathway. So as I mentioned, RIG-I in the uninfected cell is in a autoinhibitory state where its card domains are basically masked by the helicase. But when it detects the viral RNA, it undergoes a conformational rearrangement, and where the viral RNA with a motif, 5' triphosphate motif, are bound by the helicase and C-terminal domain, while the card domains are open and exposed for interaction with host proteins, other host proteins in the pathway such as MAPs. So the interaction of this card-card interaction actually activates transcription factors such as interferon regulatory factor 3, which after getting activation induce the interferon expression. So now there are many different proteins, and NS1 is known to inhibit a lot of different peripheral proteins, but we'll just focus on the interaction of NS1 with the RIG-I protein and how it inhibits this pathway. Also, okay, now coming to the first aim is to understand how NS1 interacts with this RIG-I protein, to understand the structural basis of interaction. That means to elucidate what domains of both of these proteins involved in interaction, as well as determine the binding interface, binding characteristics. Now the aim 2 is basically understanding molecular basis of strain-specific interactions. So NS1s of different strains of influenza virus proteins have different strategies that employ to inhibit host immune response. We want to, in the second aim, we want to see how the strain-specific NS1s, strain-specific mutations, basically in NS1, modulate their interaction with RIGA, and whether the binding between NS1s of different strains is different, and how does this interaction between NS1 and RIGA inhibit the oligomerization maps, which I did not mention is an essential step for activation of interferons. Okay, so coming to significance, by the end of the study, we anticipate that we will determine a structural basis of NS1 inhibition of RIGA, and that will also give us the binding interface, which could be potentially targeted by therapeutics for inhibiting NS1 itself, as well as we will learn in the second aim the molecular basis of strain-specific interaction, which would shed more light on the evolution of this viral protein NS1, as well as the viral and host interactions. Okay, so now coming to the first aim is to determine the structural basis of NS1 RIGA interaction. And people have shown that NS1 and RIGA interact, but there has not been any structural model of this interaction. So since RIGA interacts with RNA, the viral RNA, in the presence of the viral like NS1, we saw that using electrophoresis mobility shift assay, if you see on the fourth lane, we see a shift in the migration of the RIGA-bound double-stranded RNA. That indicates that there is a formation of a ternary complex of NS1, RIGA, and viral RNA. So, as a reference, when we truncate the RIGA and delete the CART domain, we do not see any sorts of shift in the migration. That indicates that NS1 directly targets the CART domain of RIGA to interact for the interaction. Now, we have established that NS1 targets RIGA. Next, our aim is to determine what domains of NS1 and RIGA are involved in interaction. So for that, I have listed down the different domains of NS1 and RIGA, and we will study individual interaction between individual domains of NS1. For example, let's say RBD, the RNA binding domain, with the CART domain of RIGA, the CART1 of RIGA, CART2 of RIGA, as well as the combination of 1 and 2. And we'll do that the same for the effector domain as well. So, and the interactions between these different domains will be studied using different methods. For example, the in vitro pull-down assay, and the observations from the in vitro pull-down assay will be confirmed, validated through using bilayer interferometry and isothermal titration calorimetry, which would give us binding characteristics. For example, what is the association rate of these domains, what is the affinity between different domains, and results from these studies would give us help us elucidate the domains which are directly involved in the interaction. Okay, so for the next aim, to get more details of interactions between NS1 and RIGA, we'll use an integrative structural biology approach to determine the structure of NS1 and RIGA complex. First of all, we'll use cryo-electron microscopy. So let's say if the RIGA is bound to double-stranded RNA with the CART domain exposed. If NS1 targets the CART domain, so the ternary complex of NS1, RIGA, and double-stranded RNA will be around 160 kilodalton. So cryo will be appropriate for the determination of structure of this complex. And we have already, using cryo determined the structure of RIGA bound to double-stranded RNA. And this image on the left is basically the 2D classification averages, and this is the cryo map on the gray. In the gray is the cryo map of RIGA bound to double-stranded RNA, which is superposition with the existing structure of RIGA. As you see that the structure matches really well, but what we observe is that the structure is basically the helicase and C-terminal domain of RIGA that are bound to double-stranded RNA. We do not observe the CART domains which are exposed to the solvent. This is because the CART domains are flexible, they are not observed in the cryo map. Now, if NS1 interacts with this protein RIGA through the card domain, what we plan to use is basically use two different reguides on the same RNA, and that helps formation of a card tetramer that stabilizes the structure so that it is visible on the electron map. And then in the presence of NS1, we'll be able to determine structure of the complex. Okay. So as an integrative structural biology approach, we will also combine many different other methods. For example, X-ray crystallography. For example, let's say from binding studies, we observe that card domain of a reguide is like the full length NS1 interacts with the card domain of reguide. We can use X-ray crystallography to determine the structure of the complex. And this will also be further aided by AlphaFold 2, where we'll take individual domains and then try to determine a structural model or predict structural model of the complex. And based on the AlphaFold 2 observations, we'll also use different NMR methods such as NMR paramagnetic relaxation enhancement to determine, for example, let's say if you label the RBD of NS1 with a paramagnetic spin label and then isotopically label the card domain, when we like if the domains interact, we'll see a quenching of the NMR signal that would give us an idea about the binding interface as well as the distance between different binding sites. Using the distance restraints from the NMR paramagnetic relaxation enhancement as well as binding interface information, we'll incorporate the information into HADOC, a molecular docking platform, to also again determine the structural model. And all of this information will be validated, the binding will be validated using mutagenesis as well as methods such as BLI and ITC. And at the end of this AIM1, we'll get a structural basis. We expect that we'll determine a structural basis of NS1 interaction with reguide. Now coming to AIM2, to understand the molecular basis of strain-specific NS1 reguide interaction, people have already studied the interaction between NS1, but one of the just the RBD domain of NS1 with the card domain of reguide, and using electrophoresis mobility shift assay, we observed the same thing. We saw that the RBD domain of NS1 interacts with the card domain of reguide in the presence of a double-stranded RNA. But if you truncate the reguide and delete the card domain, there is no such interaction. So in this AIM2, we want to focus the interaction between RBD of NS1 and the card domain of reguide, and we'll use many different strains. For example, the 1918 pandemic, strain from the 1918 pandemic, 1918 H1N1, and the Udon S3N2 and Vietnam H5N1, which is an avian influenza strain, as well as the recent California 2009 pandemic strain. And when we did sequence alignment of the RBD domain of different strains, we see many different mutations with respect to the original 1918 pandemic strain, and we think that these mutations may modulate the interaction of NS1 with reguide. And some of these mutations are conservative mutations, some of them are non-conservative mutations, which may stabilize or destabilize the interaction of NS1 with reguide. Okay, so I have pointed out the mutations in the NS1, the RBD domain of NS1s, and we see that there are many different mutations in the helix 1 and 3, which are at the opposite interface of the RNA binding domain, RNA binding interface. So these mutations may modulate the interaction between NS1 and reguide, and to basically study that, whether the interaction between different strains of RBD and reguide are different, we'll again use BLI and ITC. If they interact, also use, for example, different card domain of reguide. So in the previous study, they have only used the card 2 domain, but we'll extend it and include card 1 domain also in our study. And if the different RBD domains interact with reguide, we'll then try to find out these specific residues which are responsible for these interactions. What we'll do is basically, for example, let's say one of the mutations R67A, R67, which is mutated to tryptophan in UDOM, will graft these mutations, take these mutations and graft on the 1918, the original 1918 RBD, and then try to study the interaction between this mutated RBD with the CAR domain and see whether these residues affect this interaction. Again, we'll use an integrated structural biology approach to determine the structure of the CAR domain, the complex of this RBD with the CAR domain alone using X-ray crystallography, and we'll also include the RBD domain of other strains which interact with CAR domain. We'll again use AlphaFold 2 to determine a structural model of the complex of RBD and CAR, and use NMR methods such as chemical shift perturbation with molecular docking. So let's say this is we'll acquire basically a 2D HSQC spectra of the RBD domain of NS1, and when we add the unlabeled CAR domain of Riga, if there is interaction, we should see some changes in the or perturbation in the shift, chemical shift of certain residues which might be involved in the binding interface, and then we'll measure the changes and then plot that across the residues and see which residues have on an average higher perturbation, and then try to map those residues on the structure of the RBD to see what residues might be involved in the interaction. And we'll take these NMR CSP information as well as metagenesis informations and incorporate them into platforms such as HADOP to determine a structural model. So, and at the end of M2, we should get a molecular basis of strain-specific NS1 and Riga interaction, as well as this also should give us more information on the evolution of this NS1 protein as well as its interaction with a key viral sensor, Riga.